Hey everyone, how's it going? So this is going to be our last topic for the unit. We're going to be talking about gene mutations where bases are inserted or deleted. And the first thing we're going to talk about is when they're inserted. So I'll just explain through this example what that means. This is our regular, or not our regular, but this is the DNA sequence that we had in the last video, and we found that this was the RNA sequence and these were our amino acids. So we had our start amino acid and then some other ones and then our stop, which created a protein. So this is just the DNA strand written out again, but what I'm going to do is I'm going to insert another base in there, and we're going to see how that changes things. So just right here, I think I'm going to randomly insert a T. So now our RNA, our opposite of TAC, is still AUG. TTG becomes AAC. GCG is CGC. But now, instead of reading ACC, it's going to read TAC. Because remember, it just reads 3 in a row. It's not going to be like the original 3, but it's just going to be 3, 3, 3, and then this would be our next 3, TAC, which gives us AUG. Then our next one would be CAC, which would be GUG. And then we would have this GAC, which would be CUG. And then we just have an extra T left over. So how is this going to affect our amino acids? Well, AUG is methionine. AAC is asparagine. CGC is arginine. And now here's where our mutation is. Our next one again is AUG. So AU and G is methionine again. Then we had GUG, which is valine. And then we had CUG, so CU and G is leucine. And this is what that sequence looks like. So if we compare it to our original one, which is here, at this point here is where we get a difference. And another thing you'll notice is here we had a stop codon, but here we don't have a stop codon. So our amino acid sequence is completely different, and it produces a completely different protein. So what happened was we lost our stop signal, and all of our amino acids after that point of mutation completely changed. So that's one thing that can happen with insertion. So what we did is we inserted a letter and we lost the ending of our protein. So it's just going to keep going and going. Another thing that you can notice in the RNA sequence is the way to tell if you have an insertion is if there's like an extra letter left over at the end. So that's insertion. Now let's take a look at deletion. All right, so let's talk about deletion now. So deletion, you might be able to guess, is when one of our bases gets deleted. So insertion is when a base gets added. Deletion is when one gets taken away. So here is the same example again, the one that we've been using the whole time. Now, here's our mutated strand of DNA, and let's get rid of maybe this A here. So let's take a look and see how that changes our RNA sequence and our amino acids. So TAC is AUG, TTG is AAC, GCG is CGC. But now remember, it always reads it in groups of 3. So our new group of 3 is CCA. So that would become GGU. Then our next one would be CGA. So that becomes GCU. And then we still have this CT left over. So that would be G and A. But that's not enough to make a codon. So our amino acids, what they would look like is AUG is still methionine. AAC is still asparagine. CGC is still arginine. Now our new base, instead of UGG, we have GGU. So that becomes G, G, and U is glycine. G, C, U is alanine. And then we just have these 2 letters left over. So once again, we've lost our stop codon. And from where the mutation happens, we have a completely different amino acid sequence. So instead of the tryptophan, we have glycine. Instead of cysteine, we have alanine. So again, it leads to a completely different amino acid sequence, and it leads to a completely different protein, and we don't have a stop codon. So yeah, that's what happened there. So on your assignment, there's going to be an example like this, one of insertion and one of deletion, and it's just up to you to take a look at the differences and figure out how the mutation changed your amino acid sequence. If you have any questions, just let me know. But until next time, guys, have a good one. See ya.